Hello all, my name is Onyekweli. I'm a physician in the Department of Internal Medicine at the Obafemi Awolowo University Teaching Hospital Complex with additional research responsibilities at the Lagos University Teaching Hospital Cancer Center. Today, I want to talk about a paper I recently published in Oncoscience. The paper is titled Engineering CAR-T Cells for Solid Tumors, Overcoming the Microenvironment through Integrated Design and Clinical Translation, published in Oncoscience. CAR-T cell therapy has transformed the treatment of blood cancers. Complete remission rates in leukemia now exceed 80%. But in solid tumors, the pooled response rate across trials sits at only about 9%, almost an order of magnitude lower. This paper is a synthesis of why that gap exists and of the engineering strategies now closing it. I organized it around 4 barriers a CAR-T cell has to survive inside a solid tumor: a physical barrier of dense stroma and abnormal blood vessels, a metabolic barrier of low oxygen and nutrient depletion, an immunosuppressive barrier of signaling molecules that shut the T cell down, and an epigenetic barrier that locks it into a state of exhaustion. The review draws on over 100 studies, including 2024 and 2025 results in glioma, gastric cancer, and liver cancer, where objective response rates have reached 50 to 57%, a genuine turning point for the field. It closes with a tiered framework for how these strategies should be combined in the next generation of constructs. My own research sits at the interface of the gut microbiome, epigenetics, and immunity. I've published on how bacterial metabolites, specifically short-chain fatty acids, regulate an enzyme called HDAC2 and how that shapes downstream metabolic and immune signaling, partly through a gene called PDK4. CAR-T exhaustion in solid tumors runs through a closely related mechanism, a DNMT3A-driven chromatin remodeling program that locks T cells out of an effector state. In fact, this review itself covers that exact convergence, a section on how gut-derived short-chain fatty acids, such as butyrate and pentanoate, augment CAR-T activity through the same class 1 HDAC inhibition I work on in my own research. Seeing that link directly motivated this review. There was also a clinical motivation. In my work in oncology and occupational health here in Lagos, I see, close up, how far current treatments still fall short for solid tumors, and how much promise is now emerging in engineered cell therapies. I wanted to bring those two threads, the mechanistic biology and the clinical trajectory, into one coherent account. A few things stood out while writing this. The first is how much the field has genuinely shifted in the last 2 years, from what I call a potency era, where the goal was simply to make the T cell stronger, to a resilience era, where the goal is to protect the T cell from a hostile environment instead. A dominant negative TGF-β receptor, for instance, doesn't make the T cell more aggressive. It simply blocks a suppressive signal, and that alone has driven some of the strongest results in liver cancer. The second is how underappreciated the systemic side of this still is, how a patient's stress physiology through the hypothalamic pituitary adrenal axis can quietly work against a CAR-T cell days before it ever reaches the tumor. That's a dimension the engineering literature has mostly overlooked, and honestly, the least glamorous but maybe most important part of the process was verification. This review draws on over 100 sources, and checking every citation against its original data, line by line, took longer than writing most of the sections. But that discipline is what makes a synthesis like this trustworthy. Looking ahead, I have several threads in progress that build directly on this review. I'm finishing a paper on piezo channels and mechanosensing in oncology in an African clinical context for African health sciences, extending the mechanical side of the tumor microenvironment that this review only touches briefly, since those same channels translate physical forces in the tumor stroma into signaling changes. I also have two further reviews in active development. One looks specifically at the immunometabolic determinants of CAR-T persistence and exhaustion, going deeper into the T-cell biology this paper introduces. The other is a systematic review of NK cell therapeutics in gastrointestinal malignancies, extending the same engineering logic to a different immune effector cell. Alongside those, I'm running a primary computational research project examining a specific axis linking tumor-associated bacterial metabolites to checkpoint response. Early stage, but a direct continuation of the biology this review is built on. Longer term, I'm actively exploring doctoral and postdoctoral opportunities in tumor immunology or computational cancer biology, where I can pursue this line of research full-time. I want to thank my co-authors, Gloria Omorui, Christopher Akintayo, and Samuel Kehinde Olaniyi, along with the Obafemi Awolowo University Teaching Hospital Complex and Afe Babalola University. Thank you also to the editors and peer reviewers at Oncoscience for their careful work on this paper, and to everyone watching. Thank you.